Here we'll learn about the anatomy, histology, and physiology of the adrenal gland. To begin, start a table and denote that the adrenal gland is structurally and functionally divided into the outer cortex and inner medulla. Denote that the cortex is embryologically derived from mesoderm and produces steroid hormones from cholesterol. The cortex can synthesize cholesterol de novo, but about 80% of the cholesterol used is obtained from circulating low-density lipids. See the links in our notes for a review of cholesterol. Denote that each of the three layers of the adrenal cortex is regulated by ACTH, adrenocorticotropin, though the outermost layer, the zona glomerulosa, is primarily regulated by angiotensin II. More specifically, denote that ACTH upregulates adrenocortical cell LDL receptors and increases enzymatic activity of cholesterol side chain cleavage, which releases cholesterol from the LDLs. We address adrenal cortex hormone biosynthesis elsewhere. Denote that steroid hormones are metabolized in the liver and secreted in the feces and urine. Then denote that the inner medulla has neural crest cell origins. It secretes catecholamines in response to sympathetic nervous system stimuli. The adrenal medulla is sometimes called a specialized ganglion. Now let's begin our diagram by showing the blood supply of the adrenal gland. As we do so, notice that it receives substantial blood flow despite its relatively small size. First, show the right and left kidneys, with the adrenal glands at their superior poles. Thus, the alternative name is suprarenal glands. Indicate the aorta and renal arteries. Show the vena cava in the left renal vein. Now show that the inferior phrenic arteries arise from the aorta and give rise to the superior suprarenal arteries, which supply the superior regions of the adrenal glands. Then show that the middle suprarenal arteries arise directly from the aorta and travel to the adrenal glands. Show that the inferior suprarenal arteries branch off the renal arteries. Then show that venous drainage of the adrenal glands is asymmetrical. Show that the right adrenal gland drains directly into the vena cava, but because of its relative distance from the vena cava, the left adrenal gland first drains into the left renal vein. The venous system lies anterior to the arterial system. This is especially important when viewing axial CTs of the abdomen. Now let's learn about adrenal gland hormone secretion. We'll start with the cortical hormones, the steroid hormones, which are regulated by ACTH. First, draw the hypothalamus and anterior pituitary gland. Show that neurosecretory cells originate in the arcuate nucleus of the hypothalamus and that their axons terminate on capillaries of the hypothalamic pituitary portal system. Within the anterior pituitary, show various collections of endocrine cells. Label the corticotrophs with C, and show the nearby capillaries that deliver hormone products to the blood supply. Now return to the hypothalamus and indicate that it secretes corticotropin-releasing hormone, CRH, into the neurosecretory cells. When it reaches the anterior pituitary, CRH stimulates corticotropin release of ACTH, which then travels in the blood to the adrenal cortex. Show that in response to ACTH, the adrenal cortex releases cortisol and androgens. Return to the hypothalamus and show that hypoglycemia and stress trigger the release of CRH, whereas ACTH and cortisol provide negative feedback. We've indicated that ACTH negative feedback on the hypothalamus represents a short feedback loop, whereas the cortisol negative feedback is a long feedback loop. At the level of the anterior pituitary gland, indicate that cortisol provides negative feedback to inhibit the release of ACTH. This is the short feedback loop for cortisol. For completeness, return to the adrenal gland cortex and show that ACTH also stimulates aldosterone secretion. However, indicate that aldosterone secretion is primarily regulated by the renin-angiotensin II response to low renal blood pressure and by extracellular potassium concentrations. Thus, low renal blood pressure and elevated potassium levels stimulate aldosterone secretion. Now show that the adrenal medulla is stimulated by the sympathetic nervous system. Draw a neural axis, brain and spinal cord, and show that preganglionic sympathetic fibers from the spinal cord release acetylcholine on the chromaffin cells of the medulla. We discuss this in detail in our autonomic nervous system tutorials. Next, let's show the layers of the adrenal gland. First, from outer to inner, show the capsule, cortex, and medulla. Then show that the cortex comprises 3 sublayers, each with their own products. Indicate that the zona glomerulosa produces mineralocorticoids, the most important of which is aldosterone, which regulates salt and water balance. Thus, when you think of the zona glomerulosa layer, think mineralo for salt. Then indicate that the zona vesiculata produces glucocorticoids, the most important of which is cortisol, which regulates blood sugar. Thus, when you think of the zona vesiculata, think gluco for sugar. For completeness, indicate that this layer also produces a small quantity of androgens. Indicate that the zona reticularis produces androgens, specifically DHEA, and A4, which regulate sex characteristics. Thus, when you think of the zona reticularis, think androgens for sex. For completeness, indicate that this layer also produces a small quantity of glucocorticoids. Remember, these were primarily produced by the zona vesiculata. Next, show that the medulla, which comprises the center of the adrenal gland, produces norepinephrine and epinephrine. So think medulla is the sympathetic nervous system. Notice that we've indicated from superficial to deep, salt, sugar, sex, and sympathetic regulation. Hence, we can use the mnemonic, the deeper the sweeter, but it doesn't capture the full range of the adrenal functions. It only describes the adrenal cortex. Now let's show key histological features of these layers, which will help us understand how they got their names. Click the links in our notes to see real histological samples of these layers. Begin with the capsule and show that it comprises fibrocollagen fibers and capillaries. Then show that the zona glomerulosa comprises secretory cells with round nuclei arranged in irregular rounded nests or clusters, also known as glomeruli. Indicate that these nests are separated by fibrous extensions of the capsule. These are the trabeculae. This is the thinnest layer of the cortex. Show that the zona vesiculata comprises columns or cords, also known as fascicles, of cells separated by collagen fibers and capillaries. These cells have abundant cytoplasm, which stains pale due to the presence of abundant lipid droplets. Notice that this is the whitest layer of the cortex. Next, show that the zona reticularis comprises small branching cells that form a network, also known as a reticularis, with capillaries. Because these cells have low lipid levels, they stain dark, indicate that you may see brown lipofuscin pigments in this layer. Lastly, show the chromaffin cells of the medulla, which are arranged in clusters around venous channels that deliver catecholamines to the blood. This concludes our diagram.